Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to tackle a practice problem set that relates to our lecture on RNA and the genetic code. Let's go ahead and get started with problem number 1. Problem number 1 says, what role does peptidyltransferase play in protein synthesis? Let's work through each answer choice. A says it transports the initiator amino-acyl tRNA complex. That is not correct. The initiator transfer RNA is brought to the ribosome by initiation factors, not by peptidyltransferase. So that's not what we're looking for. B says it helps the ribosome to advance 3 nucleotides along the messenger RNA in the 5' to 3' direction. This process is called translocation, and it's actually carried out by elongation factors, not by peptidyltransferase. So again, this is incorrect. C says it holds the protein in its tertiary structure. Also incorrect. Tertiary structure refers to protein folding that happens after translation is done, but peptidyltransferase is active during translation. We're left with answer choice D, which says it catalyzes the formation of a peptide bond. And this is our correct answer. Peptidyltransferase is an enzymatic activity of the large ribosomal subunit, and its job is to catalyze the formation of peptide bonds between the amino acids that are carried by the transfer RNAs in the ribosome. So again, 1 is D. Problem 2 says, a mutation in which of the following components of the lac operon would lead to a significant reduction in the expression of lactase, one of the structural genes. Let's again work through the answer choices. A says the operator. Now, a mutation here might actually increase gene expression if it prevents the repressor from binding. So that wouldn't reduce expression. So A is not correct. B says the regulatory gene. This gene codes for the repressor protein, but if this gene were mutated and the repressor couldn't be made, then expression would go up, not down. So that's not our answer either. C says the promoter. This one is correct. The promoter is the site where RNA polymerase binds to initiate transcription. If the promoter is mutated and RNA polymerase can't recognize or bind properly, then transcription won't happen efficiently, and that will lead to a major reduction in lactase expression. Let's still work through D, structural genes. A mutation here might change the structure of the protein that's made, but it won't necessarily reduce expression unless the mutation interfered with transcription. So while it could affect function, it doesn't best explain a reduction in expression. What would is a change in the promoter. So the best answer here is C. Problem 3 says, topoisomerases are enzymes involved in blank. Now, topoisomerases are enzymes that deal with DNA supercoiling. So as the DNA is being unwinded, like during replication or transcription, the strands, they can get tightly wound or tangled. So topoisomerases come in and they relieve that tension by making some temporary cuts and then resealing the DNA. So they're going to be active during both DNA replication and transcription. So that makes the correct answer here, answer choice A. Problem 4 says, valine tRNA is the transfer RNA that carries valine to the ribosome during translation. Which of the following sequences gives a proper anticodon for this tRNA? Refer to this table. Now, looking at the codon chart, valine is coded by GUU, GUC, GUA, and GUG. All of these are codons on the messenger RNA. Now remember, the anticodon is on the transfer RNA, and it has to be complementary and anti-parallel to the codon. So if the codon is, for example, GUA, then the anticodon would be written 3' to 5' as CAU. But in most multiple choice questions like this, we write the anticodon in the 5' to 3' direction. So that means you reverse the order. Now let's walk through it carefully using one of the codons for valine. We're going to choose GUA. The base pairings would be G pairs with C, U pairs with A, and A pairs with U. So the anticodon would be CAU, written 3' to 5'. To reverse it and write it in the 5' to 3' notation, we're going to flip it, and it's going to be UAC. That makes the correct answer choice here C. 4 is C. Problem 5 says enhancers are transcriptional regulatory sequences that function by enhancing the activity of blank. A says RNA polymerase at a single promoter site. B says RNA polymerase at multiple promoter sites. C says spliceosomes and lariat formation in the ribosome. And D says transcription factors that bind to the promoter but not to RNA polymerase. Now, enhancers are special regulatory DNA sequences, and they don't code for proteins themselves, but instead they serve to increase the efficiency of transcription. Now, they do this by helping RNA polymerase bind more effectively at the promoter. The key here is that the enhancers don't work directly by binding RNA polymerase themselves. Instead, they are going to bind by specific transcription factors. And those transcription factors then interact with the transcriptional machinery at the promoter, even if the enhancer is located far away on the DNA strand. The DNA strand can then loop around to bring the enhancer and the promoter regions into contact. So if we want to say this clearly, enhancers increase the activity of RNA polymerase at a promoter, and that's exactly what answer choice A is describing, RNA polymerase at a single promoter site. Now let's rule out the other choices. B says multiple promoter sites, but enhancers are typically specific to a particular gene or promoter. So this isn't the best answer. C mentions spliceosomes and lariat formation, which are part of RNA splicing, not transcription initiation, so that is unrelated. And choice D mentions transcription factors, but says that they bind the promoter and not RNA polymerase. So that misses the point. Enhancers interact through transcription factors, yes, but the ultimate effect is on RNA polymerase activity. So again, answer choice A is correct. Problem 6 says, in the genetic code of human nuclear DNA, one of the codons specifying the amino acid tyrosine is UAC. If one nucleotide is changed and the codon is mutated to UAG, what type of mutation will occur? Is it silent mutation, missense mutation, nonsense mutation, or frameshift mutation? Now, UAC is a codon that codes for the amino acid tyrosine, but UAG is different. This is the genetic code for the stop codon. This is one of the stop codons that you have to commit to memory. That means if this mutation happens at the 3rd position, which is our wobble position, translation would stop prematurely and the protein would be cut short. Now, a mutation that changes a codon for an amino acid into a stop codon, that's called a nonsense mutation. So the correct answer here is going to be C. Problem 7 says, which of the following is not used by eukaryotes to increase the transcription of a gene? Let's go through our options. Answer choice A says gene duplication. Now, when a gene is duplicated, more copies of the gene exist, which can lead to higher expression. So gene duplication can increase transcription levels. This is not what we're looking for. B says histone acetylation. This is a well-known mechanism that will open up the chromatin structure and make DNA more accessible to transcription machinery. This is going to clearly enhance transcription, so again, not what we're looking for. C says DNA methylation. That's the correct answer. Methylation of DNA usually silences genes, so it makes the DNA less accessible by tightening up the chromatin structure, and that can also prevent transcription factors from binding. So this actually reduces gene expression, and that's why it's the correct answer. Let's still work through D. D says enhancers. These are regulatory DNA sequences that increase transcription by helping transcription factors and RNA polymerase bind more efficiently. So this increases transcription. That's not what we're looking for. So A, B, and D increase transcription, but C decreases it. And that is what we're looking for, which is why 7 is C. Problem 8 says, when a trypsin converts a chymotrypsinogen to chymotrypsin, some molecules of chymotrypsin bind to a repressor, which in turn binds to an operator region and prevents further transcription of trypsin. This is most similar to which of the following operons? A says the trip operon during lack of tryptophan. B says the trip operon during abundance of tryptophan. C says the lac operon during lack of lactose. And D says lac operon during abundance of lactose. So let's break this down. The passage tells us that when trypsin converts chymotrypsinogen to chymotrypsin, some chymotrypsin feeds back and binds to a repressor. That repressor then binds the operator and it blocks further transcription of trypsin. This is a classic negative feedback loop where the end product shuts down its own synthesis. And that's exactly what happens in the trip operon when tryptophan is abundant. When there's enough tryptophan goes ahead and binds to the repressor and activates it. And that repressor then goes ahead and binds to the operator to stop further production of tryptophan synthesizing enzymes. So the situation described in this question, where the final product inhibits its own synthesis, it most closely resembles choice B, the trip operon during abundance of tryptophan. Moving on to problem 9. 9 says, which of the following RNA molecules or proteins is not found in the spliceosome during intron excision? During intron excision, the spliceosome is the molecular complex that's going to be responsible for removing non-coding sequences from pre-mRNA. Now, pre-mRNA is another way of saying heterogeneous nuclear RNA. So we know that this is involved. We can go ahead and cross out answer choice B. Now, the spliceosome, it's composed of small nuclear RNA and proteins that form snRNPs. So we can go ahead and cancel A and D as well. The spliceosome assembles on the pre-mRNA to carry out splicing. However, this choice right here, short hairpin RNA, that's not going to be involved in the process. This functions in RNA interference, not splicing. Therefore, the correct answer is C, since it is not found in the spliceosome during intron excision. Let's move on to problem 10. Problem 10 says, a 4-year-old with cystic fibrosis is seen by his physician for an upper respiratory infection. Prior genetic testing has shown that there has been a deletion of 3 base pairs in exon 10 of the CFTR gene that affects codons 507 and 508. The nucleotide sequence in this region for normal and mutant alleles is shown below. X denotes missing nucleotides, and we have that given to us right here. What effect will this mutation have on the amino acid sequence of the protein encoded by CFTR gene. A says deletion of phenylalanine residue with no change in the C-terminus sequence. B says deletion of a leucine residue with no change in the C-terminus sequence. D says deletion of a phenylalanine residue with a change in the C-terminus sequence. And D says deletion of a leucine residue with a change in the C-terminus sequence. And here we have our codon table to reference. So if you're trying to answer this without my help first, reference this table. But let's now tackle this together. The first thing that I want to point out about this problem is that we're told that the mutation occurs in the coding strand of DNA. So that means the messenger RNA sequence is going to look almost the same, except that U replaces T. In the normal gene, codon 507 is ATC and codon 508 is TTT. Now, if we're looking at 508, TTT becomes UUU in the messenger RNA. This codes for the amino acid phenylalanine, and you can pick that up from the codon table right here. UUU is phenylalanine. Now, in the mutant version, one base from codon 507 and two from codon 508, they're deleted. And so as a result, 507 is actually going to be read as ATT, and codon 508 is just lost. It's deleted. Now, for the original 507 code, it originally read AUC for the messenger RNA. That codes for isoleucine. But after the deletion and the new way that we read codon 507, this becomes AUU. That still codes for isoleucine. So if we go to our table right here, isoleucine is right here. So AUU and AUC, they both code for isoleucine. That does not change. And that's really cool because of that wobble position. So even though the codon has changed slightly, the amino acid at that spot stays the same. The important part here is that exactly 3 nucleotides were deleted. And that means that one full codon is gone, but the reading frame, it's not disrupted. That's why the rest of the protein, including the C terminal end, it's going to be translated just like normal. What we're going to lose is just this 508 codon, which codes for phenylalanine. So this mutation causes the loss of phenylalanine residue at position 508, but the isoleucine at 507 is unchanged, and the rest of the protein remains intact. So that means the correct answer here is A, deletion of phenylalanine residue with no change in the C-terminus sequence. We can now move into problem 11. Problem 11 says a gene encodes a protein with 150 amino acids. There is one intron of 1,000 base pairs, a 5' untranslated region of 100 base pairs, and a 3' untranslated region of 200 base pairs. In the final messenger RNA, about how many bases lie between the start AUG codon and final termination codon? We're told that the protein has 150 amino acids. Since each amino acid is coded by a codon that's made of 3 nucleotides, we need to multiply 150 by 3. This gives us 450 bases. Now, it's important to recognize that this question specifically asks about the region between the start codon and the stop codon. That refers to the coding sequence, also known as the open reading frame. The 5' and the 3' untranslated regions, they don't count towards this. And neither do introns because they're going to be removed during RNA processing. So we don't have to include the 100 base pairs from the 5' untranslated region, and we don't have to include the 200 base pairs that come from the 3' untranslated region, or the 1,000 base pair intron. All we need is the number of bases that code for a protein, and that's 450 bases total. That means the correct answer here is B. Problem 12 says, peptidyl transferase connects the carboxylate group of one amino acid to the amino group of an incoming amino acid. What type of linkage is created in this peptide bond? The enzyme peptidyl transferase, this is part of the ribosome. It catalyzes the formation of a bond between two amino acids. Specifically, again, it links the carboxyl group of the existing amino acid chain to the amino group of the incoming amino acid. This kind of bond forms what we call an amide bond. And in biology, we also call this a peptide bond. It's a covalent bond that connects the amino acids in a protein backbone. And so the correct answer here is going to be amide. So 12 is B. Problem 13 says, a eukaryotic cell has been found to exhibit a truncation mutation that creates an inactive RNA polymerase I enzyme. What type of RNA will be affected by this inactivation? A says ribosomal RNA, B says transfer RNA, C says small nuclear RNA, and D says heterogeneous nuclear RNA. In eukaryotic cells, we know different types of RNA polymerases are responsible for making different classes of RNA. As for RNA polymerase I, it has a very specific job, which is that it synthesizes most of the ribosomal RNA. This is the structural and catalytic component of ribosomes. So if RNA polymerase I is inactivated, then the production of rRNA is going to be directly affected. The other types of RNA, they're made by RNA polymerase II and III, so they're not going to be impacted by this mutation. That makes the correct answer for problem 13, answer choice A. Problem 14 says, you have just sequenced a piece of DNA that reads as follows from the 5' to the 3', TC, GAGA, CATCC. What would the base sequence of the messenger RNA transcribed from this DNA be? Now, the key here is we're given the sequence of the coding strand of DNA. It's written from the 5' to the 3' direction. Notice all of the answer choices are also written 5' to 3'. So the way that we're going to do this correctly is we're going to read in this direction, and this base right here is going to translate to the first base in these sequences. Now, the difference here is it'll be read exactly the same with the complementary sequence, except we will substitute U for T. So as we read from here to here, we have C pairs with G, C pairs with G, T pairs with A pairs with T, but we will replace T with U. So that's U. Then again, we have a C right here. This pairs with G. A pairs with T, but we replace that with U. G pairs with C. A pairs with U. G pairs with C. T pairs with A, and then C with G and T with A. So the correct answer for this problem is going to be B. Now we can move into our last and final problem for this set. Problem 15 says, double-stranded RNA cannot be translated by the ribosome, and it's marked for degradation in the cell. Which of the following strands of RNA would prevent mature messenger RNA in the cytoplasm from being translated? A says identical messenger RNA to the one produced. B says antisense messenger RNA to the one produced. C says messenger RNA with thymine substituted for uracil. And D says sense messenger RNA to the one produced. So we know that double-stranded RNA can't be translated by ribosomes. In fact, when a double-stranded RNA is detected in the cytoplasm, the cell marks it for degradation because it looks like a viral threat. So the question becomes, which RNA strand could bind to the mature messenger RNA and create a double-stranded structure that would stop translation? The answer is an antisense strand. That's the RNA strand that is complementary to the messenger RNA sequence. If an antisense RNA is present, it can hybridize with the mature messenger RNA and form a double-stranded RNA molecule. This blocks the ribosome from translating it, and it signals the cell to degrade it. So the correct answer here is going to be choice B. And with that, we've completed this practice problem set. I really hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.